Scientists discover switch that helps breast cancer spread around the body. The early stage research, led by scientists from Imperial College London and the Institute of Cancer Research, London, identified a genetic switch in breast cancer cells that boosts the production of a type of internal scaffolding. This scaffolding is a type of protein, called keratin 80, and related to the protein that helps keep hair strong. Boosting the amount of this scaffolding makes the cancer cells more rigid, which the researchers say may help the cells clump together and travel in the bloodstream to other parts of the body. The researchers, who published their work in the journal Nature Communications, studied human breast cancer cells treated with a common type of breast cancer drug called aromatase inhibitors. The team found the same switch is involved in breast cancer cells becoming resistant to the medication, meaning the drugs are no longer effective if the cancer returns. Targeting this switch with a different drug could help reverse this resistance, and make the cancer less likely to spread, explained Dr. Luca Mignani, lead author of the research from the Department of Surgery and Cancer at Imperial. Breast cancer is the most common cancer in the UK, and causes 55,200 new cases every year. Aromatase inhibitors are effective at killing cancer cells, but within a decade post-surgery around 30% of patients will relapse and see their cancer return, usually because the cancer cells have adapted to the drug. Even worse, when the cancer comes back it has usually spread around the body, which is difficult to treat. Dr. Mignani said, "Up until now we didn't know the reasons behind this, but our early-stage study suggests a type of genetic switch, called a transcription factor, can turn on genes that cause the cancer cells to not only become resistant to the treatment, but move into healthy tissue around the body." This research now needs to be followed up with larger studies, but if confirmed, targeting this genetic switch could prevent cancer cells from becoming resistant to the drugs, and from spreading to other areas of the body. Aromatase inhibitors are used to treat a type of breast cancer called estrogen receptor positive. These make up over 70% of all breast cancers, and are fueled by the hormone estrogen. These cancers are usually treated with surgery to remove the tumor, followed by a course of targeted hormone therapy, usually either aromatase inhibitors or the drug tamoxifen, which blocks estrogen receptors. Aromatase inhibitors are usually given to postmenopausal women, and prevent estrogen from being produced in other tissues. Although the women's ovaries have stopped producing estrogen, some of the hormone is still made in several other tissues by an enzyme called aromatase. The medication prevents this enzyme making estrogen. However, around 30% of breast cancer patients taking aromatase inhibitors see their cancer eventually return. This returning cancer is usually metastatic, meaning it has spread around the body, and the tumors are often now resistant to aromatase inhibitors. In previous research, the same group discovered that cancer cells become resistant to aromatase inhibitors by making an alternative source of fuel. The team discovered that when aromatase inhibitors starve cancer cells of estrogen, some cells adapt by increasing production of cholesterol, which they then use for energy to survive. This means that if the cancer returns, it can no longer be killed by the same type of drugs. In the latest research, which used human breast cancer cell cultures in the laboratory, the research team found the switch that turns on genes that increase cholesterol production also activates genes that make the cells more rigid and prone to invade nearby tissues. The team also found that in women whose cancers had spread around the body, the cells contained higher amounts of keratin 80. The team say larger-scale patient studies are now needed to confirm the findings, but the research could provide new avenues for helping treat breast cancer that returns and spreads around the body. Dr. Mignani explained, although the spread of cancer around the body affects many patients, scientists are still unsure about the molecular processes that drive the movement of cells. This research sheds light on this process, and also suggests it is controlled by the same switch as drug resistance. These findings need to be replicated in bigger trials, but could potentially provide a way of stopping both drug resistance and cancer spread. Dr. Fernando Calvo, who carried out the research as team leader in tumor microenvironment at the Institute of Cancer Research (ICR), said, "Tumor evolution is one of the biggest challenges facing cancer researchers. By working out how tumors evolve drug resistance, we can find new ways to treat the cancer effectively, or even prevent resistance developing in the first place. Our study shows how drug resistance and the invasiveness of cancer cells are interconnected in breast cancer through changes in cell shape. If we understand how to block resistance, we might also be able to prevent the cancer spreading throughout the body, which would be an important step in treating breast cancer more effectively. Synthetic version of CBD treats seizures in rats. A synthetic, non-intoxicating analog of cannabidiol (CBD) is effective in treating seizures in rats, according to research by chemists at the University of California, Davis. The synthetic CBD alternative is easier to purify than a plant extract, eliminates the need to use agricultural land for hemp cultivation, and could avoid legal complications with cannabis-related products. The work was recently published in the journal Scientific Reports. It's a much safer drug than CBD, with no abuse potential and doesn't require the cultivation of hemp, said Mark Maskell, professor in the UC Davis Department of Chemistry. Maskell's laboratory at UC Davis carried out the work in collaboration with researchers at the University of Reading, UK. Products containing CBD have recently become popular for their supposed health effects and because the compound does not cause a high. CBD is also being investigated as a pharmaceutical compound for conditions including anxiety, epilepsy, glaucoma and arthritis. But because it comes from extracts of cannabis or hemp plants, CBD poses legal problems in some states and under federal law. It is also possible to chemically convert CBD to tetrahydrocannabinol (THC), the intoxicating compound in marijuana. 8,9-dihydrocannabidiol (H2CBD) is a synthetic molecule with a similar structure to CBD. Maskell's laboratory developed a simple method to inexpensively synthesize H2CBD from commercially available chemicals. Unlike CBD, there is no way to convert H2CBD to intoxicating THC, he said. One important medical use of cannabis and CBD is in treatment of epilepsy. The U.S. Food and Drug Administration has approved an extract of herbal CBD for treating some seizure conditions, and there is also strong evidence from animal studies. The researchers tested synthetic H2CBD against herbal CBD in rats with induced seizures. H2CBD and CBD were found to be equally effective for the reduction of both the frequency and severity of seizures. Maskell is working with colleagues at the UC Davis School of Medicine to carry out more studies in animals with a goal of moving into clinical trials soon. UC Davis has applied for a provisional patent on anti-seizure use of H2CBD and its analogs, and Maskell has founded a company, Syncanica, to continue development. Could better tests help reverse the rise of drug-resistant infections? A growing number of infections, such as pneumonia, gonorrhea and tuberculosis, are becoming harder to treat, as bacteria evolve defenses against antibiotics faster than we can develop new drugs to replace them. Duke University game theorist David McAdams says faster, more accurate tests for drug-resistant infections are being developed that could, at least in theory, change the game and turn the tables against some drug-resistant bacteria, but with a caveat. That's the take-home of a new modeling study led by McAdams and infectious disease expert Sam Brown of the Georgia Institute of Technology, to be published May 16 in the journal PLOS Biology. The researchers used a type of mathematical model in epidemiology called a CIS model to predict the rate of spread of resistant and non-resistant strains of a given pathogen, with and without improved testing, to see which strains would take over. Currently, a doctor who suspects a patient has a bacterial infection will often prescribe a best-guess antibiotic, without knowing for sure whether that drug will be effective. But some bacterial strains may carry genetic mutations that help them survive the standard treatment. These survivors then continue to multiply and pass on their protective genes to their offspring. Over the generations, resistant bacteria eventually push out easier-to-treat ones. A doctor can send a patient sample such as a throat swab or urine sample away for laboratory testing and adjust the prescription if necessary, but such tests can take days. Armed with rapid tests for resistance, doctors could find the best treatment for a particular patient within minutes rather than days, on the patient's first doctor visit, before they take unnecessarily broad-spectrum drugs that are more likely to fuel resistance. Knowing at the outset whether the standard first-line drug will work, doctors could prescribe that for patients when it's effective, or choose another when the traditional therapy is likely to fail. By doing so, bacteria resistant to first-line drugs lose the survival advantage they would otherwise have. It's like selectively targeting resistant infections with a sniper gun, instead of carpet bombing every infection with the same one-size-fits-all treatment, McAdams said. Not only that, doctors and others can take additional steps to reduce transmission of bacteria resistant to first-line antibiotics, such as keeping an infected child home from school longer. Diagnostic-informed treatment and control is a 1-2 punch, McAdams said. Targeted treatment takes away the survival advantage enjoyed by resistant strains, but it's the extra transmission control measures that can put these bacteria at an overall disadvantage relative to more easily treated strains. We can potentially turn the Darwinian tables on antibiotic-resistant bacteria, McAdams said. But there's a catch, the researchers found. Many microbes live for some time on our skin or in our bodies peacefully, not causing trouble, during which time these bystanders are often incidentally exposed to multiple courses of antibiotics doled out for other infections. Particularly for these types of bacteria, the team's models show, simply using rapid tests for patients who feel sick won't be enough to tip the scales. To reduce or even reverse the spread of resistance, the team found, we must also screen for asymptomatic carriers who don't show signs and usually go undetected, before they unwittingly contribute to the spread of drug-resistant germs. The stakes are high, scientists say. As many as 50,000 lives are lost each year to drug-resistant infections in Europe and the United States alone, and that number is expected to grow in coming years. According to one recent estimate, antimicrobial resistance left unchecked could kill 10 million people worldwide a year by 2050. That's one person every 3 seconds, more than cancer. Some experts warn of a grim future where even minor conditions such as urinary tract infections or scraped knees could turn lethal. Routine procedures such as cesarean sections and joint replacements could put patients at risk of contracting superbugs that existing drugs are unable to kill. The team found that only by taking swift action to identify unsuspecting carriers, treat them and keep them away from others, such as by encouraging carriers to stay home until their infection has cleared, can we hope to turn the tide. Results of their model also show that once enough infections become impervious to all the antibiotics that are available, rapid testing does little to control their spread, and new drugs are needed. To be sure, the ability to identify resistant infections faster and treat them better makes rapid diagnostics worth the investment, McAdams said. But our analysis implies that focusing on rapid diagnostics, while good, is not good enough to preserve the power of existing antibiotics. We shouldn't settle for a post-antibiotic fate, McAdams said. It's not just about discovering new antibiotics. It's about developing better diagnostic tools and intervention capabilities to preserve the antibiotics we already have. New drug could help treat neonatal seizures. A new drug that inhibits neonatal seizures in rodent models could open up new avenues for the treatment of epilepsy in human newborns. Researchers have identified that gluconate, a small organic compound found in fruit and honey, acts as an anticonvulsant, inhibiting seizures by targeting the activity of channels that control the flow of chloride ions in and out of neonatal neurons. A paper describing the research, from an international team of scientists led by Penn State researchers, appears May 15, 2019 in the journal Molecular Brain. Neonates are the most vulnerable population to seizures, but there is still no effective medication for the treatment of neonatal epilepsy, said Gong Chen, professor of biology and the Vern M. Williman Chair in Life Sciences at Penn State and the leader of the research team. The incidence of epilepsy is highest in the first year of life, with 2 to 4 infant babies suffering from neonatal epilepsy for every 1,000 live births in the United States. Unfortunately, so far there is no effective drug available that is specifically developed for neonatal epilepsy patients. Over the past decades, many drugs have been developed to treat epilepsy in adults. However, neonatal epilepsy patients are often resistant to or do not respond to current anti-epilepsy drugs, and long-term use of some of these treatments may have side effects on brain development. In the current study, Chen and his colleagues demonstrated that gluconate can inhibit seizure activity in neonatal neurons. More importantly, gluconate suppresses seizure activity in neonatal animals more effectively than in adults. This is truly exciting because we have finally identified a potential anticonvulsant drug that shows preference to inhibit neonatal seizure activity, said Chen. Gluconate is already widely used in the food and pharmaceutical industries as an inactive food or drug additive. For example, it can bind with metal ions to form stable gluconate salts, such as calcium gluconate, potassium gluconate, and zinc gluconate, that are used for the uptake of metal ion supplements. Gluconate is a small organic compound that is produced through the oxidization of glucose in many plants, fruits, and honey, said Jung Wu, an assistant research professor at Penn State and the first author of the paper. It has minimal side effects when compared to other organic ions. Because of this, our discovery of its anti-seizure function in neonates could have an accelerated path toward therapeutic development for use in the treatment of neonatal epilepsy. The research team found that gluconate inhibits neonatal seizures by targeting what are known as ClC3 chloride channels. These channels mediate a large ion current in neonatal brains but are less active in adult brains. Gluconate appears to be too large to pass through the small openings of the ClC3 channels and therefore acts as a channel blocker. We were surprised that gluconate targeted ClC3 chloride channels because they have been associated with regulating neuronal transmission, glioma proliferation, and neuron cell death, but until now, there was no mention of neonatal seizure at all, said Wu. Here we found not only that ClC3 chloride channels are highly expressed in the neonatal brain but also that they are closely related to neonatal seizures. Importantly, gluconate not only blocks the ClC3 chloride channels but it also significantly inhibits neonatal seizure activity. Its neonatal activity makes it a great specific target for neonatal anti-epilepsy drugs. In addition to gluconate, the research team also showed that a ketone body called HB, which is generated in the liver under a ketogenic, low-carb, high-fat diet, can also act as an inhibitor of ClC3 chloride channels and suppress neonatal seizure activity. In fact, ketogenic diets have been used to control some childhood epilepsies, but often with unwanted side effects in a small number of children. The similar effects of gluconate and a ketone body in inhibiting both ClC3 chloride channels and neonatal seizures may suggest a better treatment than a ketogenic diet in suppressing neonatal seizures, said Chen. Our studies not only identify a new drug target, the ClC3 chloride channels, for neonatal epilepsy but also discovered two potential anticonvulsant drugs, gluconate and the ketone body, HB, that can suppress neonatal seizures. Our work also opens a new avenue for other scientists to design even more specific and potent blockers for ClC3 chloride channels to treat neonatal epilepsy.